Hi friends and welcome back to Easy Dentistry. I've got many requests from numerous students that the exams are very near and they want to complete maximum syllabus through these videos. Friends, I'm trying my level best to complete maximum topics. I would be more than happy to know that these videos are helping you in your studies. Also, a very good luck to all for your exams, and please keep me informed how were your exams and whether these videos help you in writing the answers. Please subscribe to our channel if you have not done yet and press the bell icon to get information about our latest videos. So without wasting any time, let's start with today's topic, which is cementum. Cementum we would be studying in two parts. In part 1, we would be studying about the general features, and in part 2, we will be studying about the cementogenesis. So let's begin with part 1. In today's lecture, we would be studying about the general features of cementum, the physical characteristics of cementum, the chemical composition of cementum, the structure of cementum, various types of cementum, and the comparison between the various types. Now, starting with the general features of cementum, the cementum was first described in 1835 by two students of the scientist named Perkin. It is a mineralized dental tissue covering the roots of the tooth. It is present only in the roots of the tooth. It begins at the cervical portion of the teeth at the cemento-enamel junction and it continues till the apex. It is thickest at the apex, about 150 to 200 micron in thickness, while it is the thinnest in the cervical region, which is 20 to 50 micron. The cementum is avascular, that is, it does not contain any blood vessels. It is non-innervated, that is, it does not have any nerves in it. The physical, chemical and structural characteristics of the cementum can be compared to the bone. Now let us study the physical properties of the cementum. The hardness of cementum is less than that of bone. The cementum is less than that of the dentine. The hardness of cementum is less than that of dentine. And the color of cementum is light yellow and it lacks luster. And this yellow color is lighter than dentine. The chemical composition is the inorganic content is 45 to 50%, while the organic content is 50 to 55%. In addition to these inorganic and the organic structure, there is also presence of water. The inorganic content is mainly in the form of calcium and phosphate, which forms the hydroxyapatite crystals. Many trace elements are also present. Remember, very important point here, that cementum has the highest fluoride content of all the mineralized tissue of the body. Now, coming to the organic matrix, the organic matrix contains of collagen and non-collagenous proteins. The collagen, which is principally or mainly found in the cementum, is type 1 collagen. It is asked frequently that which is the type of collagen present in the cementum. It is the type 1 collagen. Other types such as 3, 5, 6 and 12 are also present. Now the non-collagenous proteins are BSP, the osteonectin, osteopontin and cementum derived attachment protein, which is unique only and only to the cementum. Now, what is the role of these non-collagenous proteins? They mainly help in matrix deposition, mineralization and further matrix remodeling. Various proteoglycans are also present in the organic matrix. These proteoglycans are chondroitin sulfate and heparin sulfate, while the growth factors such as TGF, FGF and IGF are also present in the organic matrix. Now, coming to the structure of cementum, the cells which form the cementum are called as cementocytes. Now, depending upon whether the cementocytes are present or absent, the cementum is divided into cellular cementum and acellular cementum. The fibers are also present, and depending on the presence or absence of fibers, the cementum is classified into fibrillar cementum and afibrillar cementum. Fibrillar cementum contains these fibers, and while the afibrillar cementum do not contain the fibers. Now, the fibers of the cementum are further divided into two types: the intrinsic fibers and the extrinsic fibers. Now, depending upon the various combinations, there are combinations of whether the cementum is cellular or acellular, fibrillar or afibrillar. There are several types of cementum. The important ones are the acellular extrinsic fiber cementum, the cellular intrinsic fiber cementum, and the acellular afibrillar cementum. Now, what is the difference between these intrinsic and extrinsic fibers? The intrinsic fibers are smaller in size. They have a diameter of only 1 to 2 micron, while the extrinsic fibers are larger and they have a diameter of 5 to 7 micron. The intrinsic fibers are parallel to the surface. They run parallel to the surface, while the extrinsic fibers are at 90 degree or perpendicular to the surface. The three major or important types of cementum are acellular extrinsic fiber cementum, cellular intrinsic fiber cementum, and acellular afibrillar cementum. There are also other types such as acellular intrinsic fiber cementum, cellular mixed fiber cementum, acellular mixed fiber cementum, and cellular mixed stratified cementum. This stratified cementum contains alternate layers of cellular and acellular cementum. Now let us study the acellular extrinsic fiber cementum or AEFC in detail. This AEFC is called as the primary cementum because it is formed first. It extends from the cervical margin up to the apical 1/3 of the root. It does not cover the apical 1/3, but it just extends up to it. The composition, it contains the collagen fibers and the non-collagenous proteins. These collagen fibers form the bulk, while the non-collagenous proteins are present in lesser quantity. Now, the extrinsic fibers of the cementum, they are perpendicular to the root surface. As shown in this diagram, it is perpendicular to the root surface. These fibers are called as the Sharpie's fibers. When cementum is thin, the Sharpie's fiber cross the entire thickness of the cementum. As shown in this diagram, the red color Sharpie's fibers, they cross the entire thickness of the cementum. But as the more and more cementum layer is laid down, this layer becomes thick, and large portion of the Sharpie's fibers, they get incorporated within the cementum. Now this AEFC is formed very slowly. So the incremental lines of the AEFC are very close to each other because of this slow rate of deposition. And these incremental lines, they run parallel to the surface as shown here. The red color incremental lines run parallel to the cementum surface. Now, in this acellular extrinsic fiber cementum, the unmineralized layer of cementum, which is called as cementoid, is absent. Single rooted teeth contain only and only acellular extrinsic fiber cementum. They do not contain any they do not contain any other type of cementum. And what is the function of this AEFC? It is anchorage, that is, it supports the tooth. Now, what is the cellular cementum? The cellular cementum is called as secondary cementum because it is formed later, and it is mainly present in the apical 1/3 area of the root. Then this cellular cementum is of two types: cellular mixed fiber cementum and cellular intrinsic fiber cementum. This cellular mixed fiber cementum is present in bulk, while the cellular intrinsic fiber cementum is very less. The cellular mixed fiber cementum is present in apical 1/3 and also in the interradicular area, while the cellular intrinsic fiber cementum is present in the apical 1/3 and also it is sometimes found in the middle 1/3 of the root. Now, how are the incremental lines in this cellular cementum? The rate of deposition of the cellular cementum is very fast. So, these incremental lines are far apart from each other as shown in this picture. The incremental lines are far apart from each other as compared to the acellular extrinsic fiber cementum. And what is the function of this cellular cementum? It is adaptation and repair. Now, let us study what are the cementocytes. The cells which form the cementum are called as the cementocytes. Suppose this is a diagram of the root. This is the tooth, and if we focus on this area of the root, we can see that this is a layer of the periodontal ligament, and the underlying cementum shows the presence of cementocytes which are present in empty spaces called as the lacunae. The cementocytes have a cell body. These are cementocytes, and these are present inside these lacunae. They have a cell body and numerous processes which extend from these cell body to form a network, and they intermingle with the adjacent cells. In the inner layers of the cementum, there are presence of numerous empty vacuoles which represent degenerating cementocytes. Now, let us compare between the acellular extrinsic fiber cementum and the cellular intrinsic fiber cementum. This question is frequently asked in exam as a long answer question. Let me tell you easy ways to remember all the points of this question. The AEFC, what is the alternate name of it? It is a primary cementum. The alternate name of CIFC is secondary cementum. Address and where it is. It is mainly found in the single rooted teeth. And in the single rooted teeth, you can never find CIFC. Where exactly in the single rooted teeth the AEFC is found? It is present from the CEJ to the apical 1/3 region of the tooth, end of the middle 1/3 region or start of the apical 1/3 region, while the CIFC is mostly present in the apical area and in the interradicular area. Now, the cementocytes, depending upon the name, acellular means the cementocytes are absent, while the cellular means cementocytes are present. Then fibrillar, extrinsic fibers are present. So, in acellular extrinsic fiber cementum, extrinsic fibers are present, while in CIFC, the intrinsic fibers are present. The next point of comparison is the function. The AEFC has a function of anchorage, while the CIFC has the function of adaptation and repair. Now, the rate of formation is very slow in AEFC and fast in CIFC. So, we have seen that the incremental lines are very close to each other in AEFC while they are far apart in CIFC. Now, let's compare the ground substance of AEFC and CIFC. The cementoid AEFC is A, that is total. Easy to remember that most of the things are absent in AEFC. Cementoid or the unmineralized cementum is absent. Non-collagenous proteins absent. Growth factors absent. The proteoglycans absent. Well, all of these are present in CIFC. Also, one more thing which is absent is the parathormone receptor present on the cementoblast, which is absent while it is present in CEIC. And also remember here that the cementoblast are derived from the Hertwig's epithelial root sheath in AEFC, and in CEIC it are derived from the dental follicle. We will be studying about this in the next part, that is cementogenesis. For now, just remember the cementoblast in AEFC are derived from Hertwig's epithelial root sheath, while the cementoblasts in the CEIC are derived from the dental follicle connective tissue. Now it is the quiz time, friends. First question: which trace element is highest in cementum than in any calcified tissue? The second question: incisors show presence of which type of cementum mainly? And the last question: type of cementum present at the apical 1/3 of roots and furcation area of the multi-rooted tooth is which type? In the next lecture, we will be studying about the cementodentinal junction, cementoenamel junction, the functions of cementum and cementogenesis. That's all for today. Please subscribe to our channel and happy studying.